Hello students, welcome to EPG Pathshala. I am Devapriya Chaudhary from Jawaharlal Nehru University, and today we are going to talk on a module called Pattern Recognition Methods in Sequence Analysis under the paper Computational Biology. This is the first part of the module, Pattern Recognition Methods. Now what exactly do I mean by pattern recognition? Now in the previous module, we saw a technique called BLAST, which could have been used to search sequences. Now that's of course not the only way. In this module, we will come across a series of different techniques of increasing complexity, I might add, which builds on the theme of sequence searching but does not do precisely the way that one saw in the BLAST algorithm. So here our searching methods can be a little fuzzy, and thereby comes the process of pattern recognition. So let us see what are the different techniques and we can go on. So why do I need pattern recognition? Common features among protein sequences can be used to group them into families, and then we can study proteins at a family level. A protein family essentially means a set of proteins which share some definite biological property in terms of a common function or maybe also a common structure. This often implies that members in a family are homologous, that is, they share a common evolutionary ancestor. Now, proteins which are very similar in structure often have similar functions but not necessarily exactly the same, and such kind of proteins often have very similar substrings within their primary sequences. So I might have a very long protein, and that two proteins with similar structures and functions need not have the entire protein similar, something which BLAST would be finding, but there might be a small stretch here and a small stretch there which might be similar, and that similarity might translate itself into a similarity in overall structure or even in terms of overall function. So what are these things called regular expressions? To understand this, let us consider the four sequences given in this figure. You can think of it as a small stretch of a multiple alignment. Now if you look at this, in the first column you have alanine or valine. In the second column you have also alanine and valine. The third column on the other hand has four different residues, while the fourth is absolutely conserved, it's a proline, and so on and so forth. So the entire information in this four sequence alignment can be encapsulated in the form of the pattern which is given below. So within square brackets, whatever is being seen refers to one column. So if you look at the first thing, it's written A or V, which indicates that in the first position you have an alanine or a valine. Same thing in the second position. But look carefully. If you have alanine in the first position, you have valine in the second position. If you have valine in the first position, then you may have alanine or you may have valine also. So in the pattern, it's just written A and V. Now if there is a correlation between what is coming in the first position and what is coming in the second position, that is not going to be reflected in the pattern. It just tells you that in each position what is the total possibility space. So in the first column, alanine or valine. In the second column, also alanine or valine, and we don't care if there are any correlations, that if it is alanine in the first position, it has to be alanine in the second, or maybe valine. No, nothing like that. Then in the third position, where it is like absolutely anything is possible, we use a character X. In the fourth position, just the opposite, where just one amino acid proline is common to all of them, so we write down that amino acid proline. In the fifth position, again it can be serine or threonine, so within square bracket we have S and T. Sixth position, again only histidine, so I just have H. In the seventh position, it's leucine, valine or isoleucine, so I have LVI, and same in the eighth position. If you look at this pattern, it already tells you something, that in the first and the second position, and also in the last two positions, you have any hydrophobic, a small hydrophobic in the first and the second, and maybe slightly larger hydrophobic in the last two positions. So given that this sort of a variation, which is very common for proteins in the family, is encapsulated in this pattern, and in the middle I have the absolutely conserved proline, the absolutely conserved histidine. There is also one position which is serine or threonine, that is those amino acids which contain an hydroxyl group, and then one position which really does not matter, whatever you may put in. So what we can see, this pattern is essentially telling the sequence space which is there at a particular position of the alignment, which in terms of proteins most likely would be a structurally equivalent position. Now one more look, we can, even as a biologist, one sees that okay, this position is hydrophobic, that position is hydrophilic, that position is that, this other chemical property, one can go and put on this kind of constraints. The pattern, as is written here, what we will call a sequence motif, encapsulates all of these informations within itself. Now given this pattern, can I search through a sequence and see, is there any subsequence within the larger sequence which matches this pattern? So here are some more examples. Look at this particular pattern. First position cysteine, then X2, 4, which means anything between 2 to 4 residues can be anything. After that, again another cysteine. After that, exactly 3 positions, which could be anything. And then one position which allows a lot of different things, mostly hydrophobics, leucine, isoleucine, valine, methionine, and tyrosine, tryptophan, etc. And then 8 positions, anything. Then a conserved histidine. Then maybe 3, maybe 5 positions, anything. Then again a conserved histidine. Any sequence that matches this pattern, right, turns out to be what is known as a zinc finger protein. Those proteins are strong binders to DNA. Okay, and there is a clear function. Now there is a database called Prosite, nowadays part of the UniProt system itself, and which started a long time ago in 1968, and they started collecting such kind of sequence patterns which corresponded to a functional protein family. And today they have a huge number of very carefully constructed sequence motifs or sequence profiles which match certain specific functional protein families. Not only that, in some cases you do not find an exact pattern or something like that. It's much more complicated. So they have defined what is called a PSSM, or a position specific scoring matrix, and which I will be describing shortly. And so they have also constructed position specific scoring matrices, and this database called Prosite is a collection of sequence motifs and PSSMs for many different functional families. Just an old example, as of 16th August 2003, this good old 14 years before today, they had 1,207 entries, which described 1,645 different patterns, rules, matrices. Today the number is much larger. So given this database, so they have made entries which is corresponding to a particular protein family or a particular characterized by a particular pattern. So let us look at it, how those entries look like. Here is an example of what is called a WW domain, a common domain family found in many different kind of proteins. So as you look at their database, you find that they have an accession number, in this particular case that's PS01159, etc., which describes the WW domain 1, and there's the other one which describes the domain 2. There is a small description which says that the WW domain, also known as RSP5 or WWP, was originally discovered as a short conserved region in a number of unrelated proteins, among them dystrophin and so on. So basically a textual description of the biochemistry of that domain. Now the thing about the WW domain is that you can recognize the WW domain by the pattern which is given there, that it starts from a tryptophan, that's the first of the W's, then you have 9 to 11 anything, then a hydrophobic, again another aromatic, then 6 to 7 anything, then GST-ANE, GST-QCR, these two groups, one more aromatic, 2 positions anything, and then a proline. Any protein thus far which matches that pattern turns out indeed by other characteristics, by other measurements, also to be a WW domain family protein. So only 9 sequences at that point of time were located in SwissProt which matched the pattern, but for other reasons could be considered as not belonging to WW domains. So therefore, this is a profile which is highly sensitive. It detects all cases of WW domains. It's not absolutely specific because we have 9 examples which it detects, but they turn out to be false positives. Any other pattern which fits it? No. So in the Prosite database, which is linked to SwissProt, so not only they give you this kind of patterns, allows you to search there, you put in your sequence of interest over there, and they will run through all the patterns and PSSLs they have, and then they will come out with a possible functional characterization of your sequence. So whenever you have a sequence and you don't know what it does, a good place to search would be to put it in the Prosite query. Just they have a text box in their web page, type in your sequence over there, and they will run through their patterns, sequence motifs, they will run through their position specific scoring matrices, and I will discuss this in a moment, and they will suggest a function. Now compare and contrast this with BLAST. In BLAST, you give a sequence, what the output comes is another sequence. It may or may not have the function which you are interested in. In this case, you are giving a sequence, what comes out is a class of other proteins which likely have the same function as your sequence. The total sequence match may not be as high as what BLAST would normally return. So here, this is a complementary approach to BLAST. So both BLAST and this one stay side by side. Again, the good thing about Prosite documentation is that they identify specific persons to go to if you have some problems with them. So in this particular case, it's Peter Bork, for example, a very well-known name in bioinformatics, who is responsible for that particular pattern. And if you have any difficulties, if you think that the pattern is not right or something like that, you can send an email to Peter Bork. Okay, to summarize, the Prosite was the very first pattern database, 1968. That's when it started. There are a few databases from that point of time which are still surviving in some form or the other. And they characterized protein families using a single motif, just one pattern. Sequence information in the motifs was reduced to a consensus or a regular expression, and then they used that initial pattern to search the entire SwissProt database by hand, which made it at that point of time very accurately curated, and they determined how many true matches, how many false matches, and so on. So if a pattern gave a huge number of false hits, then such patterns are noisy patterns. They again carefully revised so as to obtain optimal results. For some families, it could not have been possible to characterize them by a single motif. So in such cases, they used additional patterns and refined them until an optimal set of patterns could be achieved that capture the vast majority of all the members of the family, and which again then they manually annotated for inclusion in the database. The creation of the Prosite database, the history of its creation, the methods they used, I think should be optimal reading for bioinformaticians who are actually creating databases today, to show how carefully a database needs to be curated and when you are putting out to the public. We gave it some idea about position specific scoring matrices. Sometimes these are also called weight matrices. And I think very early work on this was due to Gunnar von Heine, among others, in Sweden. Now these matrices gives the relative frequency of amino acids or nucleotide bases for a DNA sequence in an ungapped alignment. So these work even in cases where the motifs are not conserved enough. Remember, in case of the motif, I am just saying alanine or valine. Now I am not saying that it's 90% alanine or 10% valine. Maybe in two different families, in some position, one of them has 90% alanine, 10% valine, the other one has 50% alanine and 50% valine. both would be quite different, but the sequence motif would not be able to distinguish. But the weight matrix can, because they are quantitatively taking into account not only which all amino acids are present in a particular position, but what is their relative proportions. Now, the matrix, of course, is of fixed length, and where intervening regions between motifs sometimes are quite useful for making matrices, etc. And this has been used to identify, for example, which are otherwise difficult to characterize protein families like transcription factor and their binding sites, which have relatively more variation than in other cases. So how are these weight matrices made? So let's look at this. So let's start with an alignment matrix from where everything comes. Let's call it Ni alpha. And we can construct a frequency matrix Fi alpha, where alpha stands for the position and the I superscript stands for the type of symbol, which could be the 20 amino acids or the 4 bases or whatever. So now for each position alpha, the frequency is given essentially the number of ith amino acid divided by the total number of amino acid. Now since some amino acid might not be even there, so you get a 0 by 0 error kind of thing. To avoid that, we usually add one pseudo count of 1, so that the 0 by 0 error is completely avoided. So we get the frequency divided at the number of ith amino acid at that position alpha plus 1 divided by the sum total of all the amino acids plus 1. Now, while this frequency is fine, so we need to normalize this frequency again, because the alignment size can vary and those kind of things can have an effect on the raw frequency. So this frequency is normalized by dividing the frequency by the background frequency of a particular character. Now let us say in some position alpha of the alignment, I have alanine coming 90% of the time. So Fi alpha is 0.9. Now, if taking all proteins, all positions, if the frequency of the existence of alanine is 90%, then this is not a surprising result, right? So, but if on the other hand, the background frequency is only 10%, then in some position where I am getting 90%, I've got a significant difference. So therefore, I need to re-weight my frequencies with the background frequency. So I do this ratio, and then of course when you do these ratios, the numbers are sometimes a little bit, they can be very close to 1.001 or 0.999. It is far better that we take the logarithm. Okay, there are mathematical reasons also why you do it, but the numbers become much more easy to handle when we take the logarithm of the ratio of these frequencies or probabilities. So sometimes we call it the log odds ratio, as you can see. So Wi alpha is the log odds ratio of the frequency of the ith amino acid at that position divided by the frequency of the same amino acid in background positions and the log of that particular ratio. Now, given a sequence as a target, I can take a window length of that, some length, say L, and then I can then see what is the log odds ratio for each position in that window length, and I can sum over that. And that would be the score of that particular subsequence with my position specific weighting matrix. So then I can move the weight matrix along the sequence and keep on scoring. So for every position of the sequence, I can get a particular score which reflects how well the local region around that sequence fits my position specific weight matrix. Now when I do this, because I'm adding up this scores at a particular position, in statistics there is something called the central limit theorem, which in this case will guarantee me that the score distribution will be perfectly Gaussian. So because the weights are now added up, there is something called the central limit theorem in statistics, which would tell us that the distribution of the score will be a perfect Gaussian. Now if that is the case, I can do some statistics to say whether a given score is significant or not, and that can simply be done by the z-score, which we first saw when we were analyzing the BLAST in the previous module. Now the z-score here is defined just by that formula, ms minus m divided by delta, where ms is the score of a particular sequence matching the PSWM. M is the average of all such sequences matching that alignment. Well, delta is the standard deviation. So when I do that, my score minus the mean divided by the standard deviation, I get the Z value, which higher the Z value, the more significant is the matching score that I get. Here is an example. Now what I have done is that I have taken a particular pattern and I have searched through sequences, many sequences, and I have gotten some score. Now remember, there will be a very small portion of sequences which truly match my pattern. That's what is likely, because I picked a protein, just maybe a specific protein with a specific function, so a specific set of proteins only would match it. And I get their scores, which is given by the shaded violet part. I also take my PSWM and run through a list of proteins, a large list of proteins, which do not match the functional family which I am interested in. These are the negatives, true negatives. When I search them, I also get a score distribution. Both of them are Gaussian distribution. Now, if these two are completely separate, then the PSW that I have created with 100% specificity, with 100% sensitivity, picks up the true positives. Unfortunately, that is most often not the case. So even my true negative distribution and my true positive distribution overlaps a little bit. If you look at that, the unshaded distribution and the shaded distribution, it overlaps in small place. So what I do, I put a threshold that anything below this is a negative, anything above this is positive. But since there is an overlap, here I will have a problem, that anything which is on the left side on the shaded curve are actually truly the sequence which should have matched, but I have decided to discard them. So these are my false negatives. On the other hand, the little bit part which is from the left distribution, which is coming to the right of the threshold which I have set, this part is actually false, but I have decided that this belongs to my family. So these are false positives. The point here is that I have to set my threshold in such a way that I make a certain balance between the false negatives and the false positives. If I take my threshold and slide it far to the left, I will never have a false negative. My method will be very highly sensitive, but I will have a lot of false positives, which indicates that I will not have a very specific method. On the other hand, if I shift it to the right, I will not have a single false positives, so which means my method will be absolutely specific, but I will lose in sensitivity. That is the part of the shaded region which is on the left of my threshold, that part I will lose. So a proper decision has to be made, or where that cutoff has to be placed, where there is a proper balance between sensitivity and specificity. So this one has to be carefully done. So given this, the false positive rate, which is the part which is coming to the right from the background distribution, right? And so we have to adjust our threshold where we can place it somewhere where the false positive rate or those which were missing, the false negatives, these are kept in manageable limits. Unfortunately, for most methods, it is not possible to get 100% in both. And so you have to make a trade-off. So somewhere you set your threshold so that both the false negative and the false positive are something which your method can handle, right? Which is within manageable limits. So in summary, the weight matrices, they take into account the position dependent probability of the residue occurrence. A sequence motif does not. Anything is present is assumed there or not there. But here, not only their presence or absence, but how frequent are they are taken into account. And this itself makes the weight matrices much more sensitive than simple sequence motifs. However, it does have one limitation. It does not take into account gaps in your alignment. That is to say, it does not take into account of indels. So iterated profile-based blast, sometimes also known as Cyblast, is a method which overcomes the problems of the gaps in our alignment, which is not being handled by PSWM. So what is this? In this approach, you take an input protein sequence and you compare with a protein sequence database using gapped version of the BLAST algorithm, which we had already discussed. The output of this is then used to construct a multiple alignment and then a profile or a position specific weight matrix from any of the significant local alignments that are found. So the original query sequence, it serves as a template for the multiple alignment as well as the profile, whose lengths are now identical to the query. So you make the profile out of the local alignments which are returned by BLAST. Having made that particular profile, you can then use this profile to search the protein database seeking for new local alignments. Now, doing all this, it turns out that you don't need to change the original BLAST algorithm very much. Small changes, tweaks here and there, and it does it. So again, let me repeat the process. You first run an ordinary BLAST, take the output, construct a multiple alignment, make a position specific weight matrix, and then run that position with a specific weight matrix to find out more examples. The BLAST algorithm itself then is used to find out the statistical significance of these alignments. And since the profile substitution scores are constructed on a fixed scale and the gap scores remain independent of position, the statistical theory which was developed for the gapped version of BLAST alignments remain applicable to this sort of profile alignments as well. So what we are doing out here, we are essentially combining the strengths of the PSWM approach and the strengths of the BLAST algorithm and its statistical significant search and all this. By doing so, not only we are handling gaps, we are also giving better statistics. So at the end of the day, Cyblast iterates the whole step arbitrary number of times until the profile scores converge. And then it calculates the profile statistics, and so which then can be used to find out the statistical significance of the hits that are generated. Now, because of this combination of position specific weight matrix and the already powerful BLAST algorithm, this algorithm Cyblast turns out to be much more sensitive than ordinary version of BLAST and not more difficult to use because everything else is built in within the algorithm, within the program itself. To understand how the Cyblast statistics are generated, we go back to the probability distribution of BLAST hits, which as we had seen before is given by the extreme value equation e to the power km e to the power minus lambda x. As we saw in the previous module, that the probability distribution of BLAST scores, so the probability that the BLAST score S is greater than some threshold x, is given by the extreme value equation e to the power minus km and e to the power minus lambda x, where again k and lambda are the scale parameters which could be found out by simulations, and it turns out that for every scoring matrix there are different values. So for the Blossom matrix, k is 0.035 and lambda is 0.252. So given that and fitting it into the extreme value equation, one can find the probabilities. So if I plot this, the extreme value distribution looks like a Gaussian kind of distribution, but the tail is now a little bit heavy. That's about the differences. Now for ungapped local alignments, okay, I can define an amino acid substitution matrix, which is something like the log odd score. In fact, it is the log odd score, where Qij is the substitution probability of amino acid I to amino acid J, Pi and Pj are the corresponding background frequencies. Now this, for normalizations, is divided by the lambda parameter. In Cyblast, each column has its unique set of target frequencies, which can be constructed into the same scale by that formula. Si is ln Qi by Pi, where Qi is the particular frequency of the amino acid I, Pi is the corresponding background, which is scaled over by lambda u, and I get the Si is the relative weight of the PSWM which is generated. So the point out there is that using the same value of lambda, question is whether it is valid. And in fact it seems to be so, because it turns out to be as an affine gap penalty model. Again, remember during the multiple alignments we had discussed these different gap penalty models. So this hypothesis was tested in a number of cases, including for example in the influenza virus hemagglutinin, and was found to be reasonably true, with a lambda has a value of 0.3176 as against 0.252, which was expected for an ungapped random alignments. So this kind of experiments generally led to the statistical theory, which applies to BLAST as well as applied to Cyblast. Therefore, the same statistics which BLAST is doing does not need serious modification for Cyblast.